Hi, welcome to our tutorial. This video is of descriptive class on basics of cell biology, precisely cell membrane structure. Cell membranes are crucial to the life of the cell. We are going to make series of tutorial videos on this chapter as playlist. If you are new in this channel, please explore our other videos. If you like, then please subscribe and hit the bell icon for further notifications. Let's learn. The plasma membrane encloses the cell, defines its boundaries, and maintains the essential differences between the cytosol and the extracellular environment. Inside eukaryotic cells, the membranes of the endoplasmic reticulum, Golgi apparatus, mitochondria, and other membrane-enclosed organelles maintain the characteristics differences between the contents of each organelle and the cytosol. Ion gradients across membranes established by the activities of specialized membrane proteins can be used to synthesize ATP, to drive the transmembrane movement of selected solutes, or, as in nerve and muscle cells, to produce and transmit electrical signals. In all cells, the plasma membrane also contains proteins that act as sensors of external signals, allowing the cell to change its behavior in response to environmental cues, including signals from other cells. These protein sensors or receptors transfer information rather than molecules across the membrane. All biological membranes have a common general structure. Each is a very thin film of lipid and protein molecules held together mainly by non-covalent interactions. Cell membranes are dynamic, fluid structure, and most of their molecules move about in the plane of the membrane. The lipid molecules are arranged as a continuous double layer about 5 nanometer thick. This lipid bilayer provides the basic fluid structure of the membrane and serves as a relatively impermeable barrier to the passage of most water-soluble molecules. Protein molecules that span the lipid bilayer mediate nearly all of the other functions of the membrane, transporting specific molecules across it, for example, or catalyzing membrane-associated reactions such as ATP synthesis. In the plasma membrane, some transmembrane proteins serve as structural links that connect the cytoskeleton through the lipid bilayer to either the extracellular matrix or an adjacent cell, while others serve as receptor to detect and transduce chemical signals in the cell's environment. As would be expected, it takes many different membrane proteins to enable a cell to function and interact with its environment, and it is estimated that about 30% of the proteins encoded in an animal cell's genomes are membrane proteins. Lipid molecules constitute about 50% of the mass of most animal cell membranes. All of the lipid molecules in cell membranes are amphiphilic, that is, they have a hydrophilic or polar end and hydrophobic or non-polar end. The most abundant membrane lipids are the phospholipids. These have a polar head group and two hydrophobic hydrocarbons tails. In animal, plant, and bacterial cells, the tails are usually fatty acids, and they can differ in length. One tail typically has one or more cis double bonds, that is, it is unsaturated, while the other tail does not, that is, it is saturated. Each cis double bond creates a small kink in the tail. Differences in the length and saturation of the fatty acid tails influence how phospholipid molecules pack against one another, thereby affecting the fluidity of the membrane. The main phospholipids in most animal cell membranes are the phosphoglyceride, which have a 3-carbon glycerol backbone. two long-chain fatty acids are linked through ester bonds to adjacent carbon atoms of the glycerol, and the third carbon atom is attached to a phosphate group, which in turn is linked to one of several different types of head group. By combining several different fatty acids and head groups, cells make many different phosphoglycerides. Phosphatidylethanolamine, phosphatidylserine, and phosphatidylcholine are the main ones in mammalian cell membranes. Another important phospholipid called sphingomyelin is built from sphingosine rather than glycerol. Sphingosine is a long acyl chain with an amino group and two hydroxyl groups at one end of the molecule. In sphingomyelin, a fatty acid tail is attached to the amino group and a phosphocholine group is attached to the terminal hydroxyl group, leaving one hydroxyl group free. The free hydroxyl group contributes to the polar properties of the adjacent head group, as it can form hydrogen bonds with the head group of a neighboring lipid with a water molecule or with a membrane protein. Together, the phospholipids phosphatidylcholine, phosphatidylethanolamine, and phosphatidylserine and sphingomyelin constitute more than half the mass of lipid in most mammalian cell membranes. In addition to phospholipids, the lipid bilayers in many cell membranes contain cholesterols and glycolipids. Eukaryotic plasma membranes contain a spatially large amount of cholesterol, up to 1 molecule for every phospholipid molecule. Cholesterol is a sterol. It contains a rigid ring structure to which is attached a single polar hydroxyl group and a short nonpolar hydrocarbon chain. The cholesterol molecules orient themselves in the bilayer with their hydroxyl group close to the polar head groups of adjacent phospholipid molecules. The hydrophobic and hydrophilic regions of lipid molecules behave in the same way. Thus, lipid molecules spontaneously aggregate to bury their hydrophobic hydrocarbon tails in the interior and expose their hydrophilic heads to water. Depending on their shape, they can do this in either of two ways. They can form spherical micelles with the tails inward, or they can form double-layered sheets or bilayers with the hydrophobic tails sandwiched between the hydrophilic head groups. Being cylindrical, phospholipid molecules spontaneously form bilayers in aqueous environments. In this energetically most favorable arrangement, the hydrophilic heads face the water at each surface of the bilayer, and the hydrophobic tails are shielded from the water in the interior. The same forces that drive phospholipids to form bilayers also provide a self-healing property. A small tear in the bilayer creates a free edge with water. Because this is energetically unfavorable, the lipids tend to rearrange spontaneously to eliminate the free edge. The prohibition of free edge has a profound consequence. The only way for a bilayer to avoid having edges is by closing in on itself and forming a shield compartment. Around 1970, researchers first recognized that individual lipid molecules are able to diffuse freely within lipid bilayers. The initial demonstration came from studies of synthetic lipid bilayers. Two types of preparations have been very useful in such studies. The first one is bilayers made in the form of spherical vesicles called liposomes, and the next one planar bilayers called black membranes formed across a hole in a partition between two aqueous compartments. That's it for today's class. We are not finished it here. For better understanding, we will continue elaborative tutorials regarding this chapter. We'll be back soon. If you like this video, please put thumbs up and do not forget to comment regarding this chapter or your any kind of suggestion for betterment of this channel. Thanks for watching. Stay with us.